answer 35 questions, each of marks to 70. The total question count is 50. Okay. In group C, you have to answer 25 questions, 4 marks each, 100. Total marks 100, and 75 questions are there. Now if you see here, CSIR NET is not only difficult as the questions are difficult, because there are in some cases questions are easy also, but the problem is about choosing the questions, because it not only check what you've learned, it also check whether you choose the correct question or not, your decision power, your selection capabilities, and your patience of reading through all the questions. So all these things are important to qualify NET. Not only studying will help, that's I'm telling you again and again, only studying will not help. Okay, and this is where you need a coaching, or this is where you need a guidance. Okay. Yeah, I'm telling you about the group C. I'm telling each of the groups here. Now you see, as you hear at the group A, or the
animal physiology,
A, it is
8 is very important. Genetics. Genetics means classical genetics as well as modern genetics. That means it will include the Mendelian, non-Mendelian genetics, as well as recombination, repair, as well as the interactions of genes, epigenetics, and all these things, whatever thing is present there. 9 is a small chapter, but they ask 1 or 2 questions from 9 every year, so you can study that. It is the biogeographical realm of India, and the distribution of animals in the different places in India, and flora and fauna of different places and stuff. Okay. 10 is the environmental biology and ecology. Yeah, biological zones. 11 is evolutionary biology. 12 and 13 biological techniques. Okay. Biology, and this part, plant biology, you'll also get 5 6, animal 4 5, and these 4 topics whichever we see earlier, because most of this biochem stuff is mathematical. Okay, the Gibbs free energy stuff, the problems from those parts and all the stuff. Okay. Now as you don't all have this list, I can't explain it right now. Next day I'll send you the list, and I can tell you from that. The course is designed in a way so that we have the normal classes, discussion classes for each of the modules. Okay. Now in those classes I will explain those topics for making the concepts more clear. Okay, this is the fine brush up again, once again. Okay. And I already sent you all the books, ebooks that we are going to follow for the modules. There are some difficulty for you to download from those books. Actually what happened, I uploaded 13 ebooks in my drive, and I sent to all of you the link, and the link I shared, I allow you as can edit. Okay. And what happened, you know what happened, after several hours, when I open my drive, I see none of the books are there, it's all gone. Okay, that's why after some time, those who tried to download the books later didn't find any book there, because I don't know how it's deleted. Someone definitely cut that book from there, I guess. Okay, so that's why I don't want to give any edit options anymore. Next we also send you the links for the books where you can download the links. Okay, so if you have any problems, if you don't understand, ADP is allosteric inhibitor and alanine is an allosteric modulator of PK. So 4 facts are provided, and they combine them. This is the type of question you are going to see more often in the group C, the cumulative question, combinatory question. Okay, they will tie 2 or 3 different things. They'll give you some statements, then they will punch the statements and then ask you the questions that which combination is true. Okay, now in this case, what you need to know, you need to know about the regulation of PK, the regulation of pyruvate kinase. So this is a fact that you need to know. So once you know that fact, then you can answer, but again nothing to analyze in this question. Okay, if you know that thing, if you know how PK is regulated, you can answer the question. Okay, for example, ATP is an allosteric inhibitor of PK, The answer, yes, it is allosteric inhibitor. Fructose 1,6 bisphosphate in an activator of PK. Yes, it is activator of PK. We know that. So these are the facts you need to know to answer the question. ADP is an allosteric inhibitor. No, it's not possible. If ATP is an allosteric inhibitor, ADP will not inhibit. It's a common sense. So definitely not. Alanine is an allosteric modulator of PK. It's true. So A, B, D, these three things are true. So which of these are true? One, not two. A, B, D. So option 2 will be the correct one. Okay. Let's move on to the next type. This is called the factual questions or analytical. In the analytical type, there are two types of analytical questions: analytical factual, and there are different pure analysis questions. This is analytical factual questions. Analytical factual means the question will imply your analysis, but you need to know some fact to imply, and then you can analyze the question. For example, a culture medium contains two carbon sources. One is preferred over another. Let me take a color. One is preferred over another, that is the glucose, and second is the lactose. Okay, which one below is the correct regarding the nature of growth curve of E. coli? You all know E. coli favors glucose. Why not? It's a simple form of sugar. Lactose is complicated form. So obviously, until and unless glucose is depleted from the medium, E. coli will not uptake lactose. We all know that, right? So growth curve will be the same as when growth is presence is only glucose. Obviously not, it will not be the true, because what happens actually, if you look at here, the lag phase, it will bring the glucose one until and unless the glucose is finished, it will not begin the curve for lactose. So it's not true. Growth curve will be same as when grown in presence of only lactose. It's again not also true, right? Because glucose is present, so obviously it has some effect there, right? Now a lag phase will be observed between the two exponential phases. Now that sounds kind of valid. So that means we can see a graph like this. This is a lag phase, this is the exponential phase, this is exponential phase. So the idea is there's a lag phase for the glucose, and then slowly start uptaking glucose, and then there will be a lag phase there in the middle. For a while there's a lag phase, and the lag phase is why, because they are not taking, the glucose is depleted, so lactose will start being utilized. Okay, so as the lactose start being utilized, there will be a lag phase observed in the middle, and then there will be again there will be the exponential phase for the lactose breakdown, and then it will go on like that. Okay. Now another thing, yeah, Miral said it's 4. Why you think it's 4? So as you see this question, it is 3. As you see in this question, you need to analyze as well as factual. Why it's factual? Because you need to know how E. coli graph looks like.
Okay, I'm back with a new other alternate connection. So can you hear me now? Okay. Okay, so let's talk. The activation of cytotoxic T cell occurs when a cell is undergoing any sort of problem. Let's say a body's cell, that is say a normal human body cell, and that cell is infected by a virus. A viral particle inserts their genetic material inside. They start making all the stuff to package the virus. And at that condition, that cell is infected. The cell wants to showcase this infection to the other cells, the immune system cells, so that this mess can be taken care of. Now the idea, when the cell is infected in such a way so that it cannot eradicate the source for the infection, in that case it's a better idea to kill that cell completely. So cell will undergo here a kind of cytotoxic feature, and that feature will be brought about by cytotoxic T cells. And how? So once they're affected, they start breaking down the components of the infective agents like bacteria or virus, whatever it is. and they will bring this part of the infective agents and load them onto a complex known as major histocompatibility complex 1 or MHC class 1. Now MHC class 1 is found in all nucleated cell in our body, and it's a process for natural combat against infective agents. So once the portion of the bacterial body is loaded onto the MHC1, it is showcased outside. Now a T cell, that is a cytotoxic T cell or TC cell we also call it, it will recognize this MHC1 complex. Once it recognizes the MHC1 complex, the cytotoxic T cell will start secreting two types of molecules. Mainly they start secreting a packet. The packet contains different enzymes, the protein degrading enzymes and hydrolyzing enzymes, and some of the enzymes are also create pore, right? So here two types of enzymes, perforins and granzyme. Two types of enzymes are there, yeah, perforins and granzymes. So perforins, what perforins do, perforins start creating pores in that infected cell, and as perforins create pores, granzymes can take entry through that pore, and granzyme can go inside and start degrading all the protein components inside the cell, which will ultimately leads to the destruction of nuclear lamina, and the cell gets destroyed. Okay, now this is what we saw is the activation of cytotoxic T cell, but there is another type of cell known as T helper cell. So how T helper cell gets activated? Now T helper cells are required. T helper cell will not kill a cell directly, but in turn T helper cells will sense the scenario, and they can activate macrophage. They can also activate other phagocytic cells in body, as well as they can also activate and mature and help in the maturation of B cells. So which is also very important. Remember I told you about the crosstalk between the T cell and B cell, so that the B cell can start developing specific antibodies against that infection. So what happens here, instead of MHC class 1, we have a different MHC, that is MHC class 2. MHC class 2 is only found in specific cells in our body, which are known as antigen presenting cells or APC. And there are few cells in our body act as antigen presenting cell. One of that is macrophage, one of that is the B cell, one of that is those dendritic cells. So what they can do, they will start engulfing those, let's say it's a bacteria, they start engulfing that bacteria. The idea is the same. They will break down the bacterial body components, because those components will act as antigen. Then they load those fragments of the bacterial body into the MHC2 complex, and that MHC2 complex is attached and fused with the cell membrane, so that that MHC2 complex can provide that antigen outside, okay, so that T helper cell can recognize. Now two types of T helper cell can recognize, either T helper 1 or T helper 2. If T helper 1 recognize that MHC2 along with the antigenic determinants there, in that case that T helper 2 will sense and it will further activate the macrophage. Many macrophage will be brought, and T helper 1 also starts secreting some interleukins, which will bring so many other phagocyting cells in that area. On the other hand, if T helper 2 gets activated, normally T helper 2 remains engaged with the B cell, because B cells are already found, and I told you early day that B cell also sense the presence of bacteria outside. And how exactly B cell engulf those bacterial cells, right? With which receptor? Tell me. I told you earlier, there is a receptor that is commonly found in surface of B cell, which binds with the bacteria or any sort of infective agent, and then they will engulf. So what acts as a receptor for B cell to engage with the bacteria? Can you tell me? Anyone? I told in the last class. If it's a B cell, yeah, it is antibody, but I want to know the name of the antibody. I wanted to know the name of the antibody. Definitely it's an antibody. You can see that thing by looking at the structure here. We have an antibody attached at the surface. But what antibody acts like this B cell receptor? IgG. No, not IgG. Not IgG. It's wrong. Why? IgG is a circulatory antibody which is converted later IgM, right? Prachi gives the correct answer, as well as this one. IgM is that antibody which is found, which is present normally because this is the first antibody that is produced. Remember, it's the antibody which is present at the early time without any modification of the antibody producing genes. It's present on the surface, IgM. Now once IgM is interacting with that bacterial component, then it will engulf it, and then it will break them down and showcase outside with the help of MHC class 2. Okay, so now T helper 2 cell can recognize that MHC class 2 along attached with that antigen, and T helper 1 will further activates B cell. Now depending upon the type of antigen that is, the T helper 2 provides the interleukin signals that will tell the B cell what kind of antigen is there, and B cell will develop and grow and mature in that way. Now if there is a specific antigen, there is a specific epitope of that antigen, so B cell need to develop an antibody which will be very specific to bind against that antigenic determinant of that foreign particle. Okay, and they will do that, and then only those genes that are present in the B cell epitope are the antigenic determinants that are present on the side. So the antibody, wherever the antibody attaches with the specific section of a protein of the antigen, is known as the epitope. That side, okay, that section is known as the epitope. The type, the characteristic nature of it is known as the epitope. Now one antigen can have multiple epitope regions and those multiple epitope regions will bind with multiple types of antibodies. So once they sense this, then they can undergo the internal changes, the modifications of the gene by the recombinations, and due to the recombination they will change, making different antibodies like IgG, and also depending upon the type of epitope they are dealing with, they modify the antibody so that the antibody can bind with the specific region of the antigen. Okay, so if you can't hear, the people who can't hear, try rejoining. Leave the class and try rejoining. Most of the people can hear okay right now. Okay. Now we'll focus on the disorders of immune system. Okay, what kind of problems we can associate with immune system. There is no receptor on surface of antigen. Receptor is present on the surface of cells like T cell, B cell. Okay, there are characteristic structures. If it's a protein, there are specific Good evening, everyone. Hope you can hear me. So, am I audible? Okay. Okay. I don't know whether everyone gets the message of today's class or not, because I declared it in last class only. So those people who only rely on the recorded classes, if they saw the recorded class earlier, then only they can join today. So actually the reason I'm taking these classes, I told you earlier even, if you have that course calendar to your hand, you will see that the dates between 23rd March and 26th March, these two dates declared as a holiday kind. Like I'll give you a assignment to do for this 1 week period, because I won't be available. That's why I put that thing in the course calendar. Now it's supposed to be that date 30th and 26th of March, but now the plan is little bit changed, so I won't be available from 25th to 10th, 25th February to 10th of March. So there is again a 1.5 week window over there. So earlier there are only 2 classes that was uh, not scheduled. So now that becomes 4 classes in between. So what I am doing here, according to the way the course is organized and the calendar is organized, 2 classes are left earlier blank. So I can do blank 2 classes. So I need to take 2 more classes for this filling this gap, because there are 4 classes gap. I need to fill that. So I'm taking 2 more classes for that. And the 2 extra classes I'm taking, one is tonight, that is 14th Feb, another one is 21st Feb. If you look at the calendar, 21st Feb is allotted for a mock test on module 3 and module 4. Okay, so what we do here, we are taking another class on 21st Feb, another group A, probably group A class, no, I think the genetics class, module 8 class. So what we do, those 2 Sundays are extra classes that I am taking, and the mock test that was supposed to be held at the 21st February will be scheduled on this some date between 25th to 10th March. Okay, so I'll give you the date. And previously there should mock test was only for module 3 and 4, but there are multiple questions, so I increased the questions of both the modules. So now there will be 2 separate mock tests, one separately on module 3 and one separately on module 4. So one of them will be conducted on both of them conducted on the dates between 25th February and 10th March. Okay, so that is the idea. I hope you understand how it's organized now. I'll give you the date for the mock test on module 3 and 4, both some are in the range of 25th and 10th. Okay, and there will be regular class on 23rd and 26th March. Okay, so that classes will be normal now. So there is a little bit change in those in that course calendar. Okay, so now today we'll discuss about group A, and many of you fear group A, and there are so many speculations about group A. The thing is, group A consists of, I told you, 20 questions there, and you need to answer 15 questions. Each questions carry 2 marks, so 30 marks allotted for group A. Now the thing is, group A means general science or general aptitude region, aptitude and reasoning, as well as data interpretation group. Now this part is compulsory, I mean common, not compulsory, you can say it's common for all the nets, like chemical science, physical science, life sciences. In all these cases, this part is kind of same, because because the idea of net exam is to figure out whether a person is qualified for the PhD, okay, for the analytical part. So that's why they check your aptitude in that case. Now if you appear for any of those government exams, any of the exams like bank, PO, rail, or any of the exams, you'll know that this part is common in all competitive exams now that is present. Although in case of net, the percentage of mark carried by this group is very less, that is 30 out of 200. On the other hand, in other exams, in other government exams or other exams, this part carries a lot of a lot marks and higher marks. Okay, now most of you fear this part because you thought that it carries mathematical questions and puzzles, and actually it carries some mathematical questions, but it's nothing like that all the questions will be mathematical or arithmetic. Okay, not all the questions are arithmetic. There are few questions asked from arithmetic aptitude. There are different zones, like arithmetic aptitude is a part where there will be questions directly associated with mathematics. For example, they can ask you questions related to profit and loss. They can ask you questions related to time and distance, time and work, age related problems, height and distance problems that we are supposed to answer. I mean, we already done that in our standard 9, 10, 11, 12. So we have already covered all those things. Those topics are repeated there, and the question levels are also those +2 level questions, but they are not very much complicated. They are based on simple linear questions from there. They also provide questions from different types of surface area related problems, like surface area volume and all these things, the problems that come from the mathematic part. This is one side of the group A, not in the entire group A, I'm telling you. The other side of the group A is puzzles. They ask puzzles. Puzzles could be consisting of so many different things. I mean, they can vary this stuff. Okay, puzzles means that could be the relationship problems between different A, B, C, D. There will be relationship problems. There could be problems related with say different time. I mean, different sort of, like if you check the question, the last question, the December question, you will see the number. They wrote hidden numbers in sentences, and you need to find that out. Those are the puzzles they ask mostly. So those puzzles are another part, and there is also reasoning is another part. Reasoning means where they ask some questions where you need to apply your common sense to answer some questions. It is actually, there are some questions, pictorial representation of questions, those are required to guess your IQ. So those IQ questions also being asked in this part. So many different varieties of questions are being asked, and there are only 20 questions and you have to answer only 15. Now the idea is, though there are, let's say among the 20 questions they ask arithmetic questions or directly mathematical questions, almost like 7-8, not more than that. Rest of the question they ask goes for the common general questions, like if you apply your common sense you can answer some of the questions. The puzzles you can solve. It is based on how much you practice some questions and how you look for different problems and problem solving approach they want to study. So those are the questions they will ask. So it is always a better idea to go for group A for at least answering few questions. Okay, because let's say even if you are not very much prepared for the arithmetic problems and arithmetic questions with all the formulas and stuff, if you forgot about them and if you don't prepare that very well, in that case also you can appear for it, and you can take at least those questions like puzzle questions or IQ related questions, you can answer them. I mean, if you look at the question papers, what value will get? Do you have any calculator? Can you calculate the value? If we calculate like 5, it will be 2, and 25, 9. 9 into 25 by 10, 45. Okay, 45 kilometer per hour. 45 kilometer per hour. That is the relative speed. Remember, it is not the direct. It's the relative speed. This is not real speed. So how we calculate the real speed now? Now, as we know the speed of the train and the speed of the person, and they are moving in the same direction, what is the formula? We need to see the difference between them. And what will be the difference? X minus 5. Let's say the actual speed of the train is x. No. Why 40? The speed of the actual speed of the train. Let's say the actual speed of the train, we know. The speed of the train is x kilometer per hour. Let's say we don't know, this is not the actual speed. The difference of the relative speed will be the difference between the actual speed of the train minus the actual speed of, I mean, the actual speed of the train minus the actual speed of the man. It will be the relative speed. We know this relative speed. The value of relative speed is 45. The actual speed of the train we don't know. Let's assume it's x. The actual speed of the man is 5 kilometer per hour. We know it's 5. So what is the value for x will be? 45 plus 5 will be 50. Okay. And why you telling 40? Because 14 is not even in the options. So it is 50 kilometers per hour. Okay. Let's move to the next one. Two trains running in opposite directions cross a man standing on the platform in 27 seconds and 17 seconds respectively. Two trains moving in opposite directions. A man is standing somewhere. The train number 1 is crossing them in 27 seconds, so take more time. The second train took only 17 seconds, so second train is faster. Remember, respectively and cross each other in 23 seconds, and they also cross each other in 23 seconds. The question is the ratio of the speed between these two trains. What will be the ratio? Now in these conditions, when you don't know the speed of any of the trains, it's kind of hard to think of the question. So in the straight way, you just take, let's say train A and train B. The speed of train A, let's say x meter per second, and for B speed is y meter per second. Now in most of the math, we never took two variables like x and y, because we know we can't calculate for two, we only take one. But here, as we are finding the ratio, we can take two, because for the ratio that doesn't matter, we can take two. So we take, one is x, one is y, the speed, we choose the speed respectively. So the length the first train carried, the first train forms, what will be the length? How we calculate the length? How will calculate the length? The length is calculated by multiplying the time taken into the speed, right? The time taken here is 27, speed is x, 27x meter. On the other hand, this is for A, for B 17y meter, that is the length traveled. Okay. Now there is another thing I did not mention earlier in the earlier formula. Now it's time to mention that. You see here the formula number 8 and 9. If two trains of length a meter and b meters are moving in the opposite direction at speed of u and v meters per second, then the time taken by the trains to cross each other would be addition of the length traveled divided by the addition of the speed. And if the two trains are traveling in the opposite in the same directions at the speed of u meters per second and v meters per second, in that case the time taken by the first train to cross the slower train would be addition of the values of the length divided by the difference between their speed. So we belong to the second format, this one. What we do, how to figure out, because you know this question is a little bit complicated. Why? Because it is telling us three things. It is telling us the time taken to cross the person, which we can easily think and calculate, we made the values. But the second thing is that they cross each other at 23 seconds, and we know they are not traveling at the same speed, they're traveling at the different speed. The train B is faster than train A. So what we need to do, the formula is to calculate the time taken to cross each other, that is here 23, the time taken to cross each other. The formula, it is equals to, in this case, the length of those trains, the addition of the length, and that addition is 27x for A, 27y for B, divided by the difference between their speed. So the higher speed train is this one, B, the difference between the speed. Here what we are talking about, we are talking about they are moving in the opposite direction. So what we do, we add things. If they are moving in the same direction, we subtract things. Here, as they are in the same opposite direction, we'll add things. So their speed, we'll add their speed. What was their speed? Taken as x meters per second, y meters per second. So we'll add them. So this is the equation that we get. Now just solve this equation. 23x plus 23y equals to 27x plus 17y. Then subtract the same thing, like that will be 6y equals to 4x, I guess. So x by y equals to 6 by 4, right? It will be 3 by 2. So the answer will be option B. Okay. Question number 3. I think it's 3 or 4. Question number 3, I guess. Na? Yes, 3. A train passes a station platform in 36 seconds, and a man standing on the platform in 20 seconds. If the speed of the train is 54 km per hour, what is the length of the platform? The idea is the train is passing a standing man, passing a station or platform in 13 seconds. This is say the platform passing in 36 seconds. And a man is also standing on the platform, passing this man on 20 seconds. Okay, check some other questions, other puzzles also, 1 or 2 more. Let's solve some other series related problems today and then end. Okay, so let's say a problem JD KF gap PM TR. What about this one? Now these are entirely IQ related problems. Now whenever you have combinations of alphabet or alphabet with numbers, in that case there will be different approaches to solve them. Anyone? One question I got, LH. How do you get LH? Everyone is saying LH. How do you get? Now it starts like, let's divide them in different groups. J, D, and then here is K, here is F, here is P. We don't know here is P, here is M, T, here is R. So what is going on? J, there is a +1 K, and D +2, right? And then if you get the same manner, so +1, so it should be L. If we get here +2, H, right? Now get +1, it should be M. But here is P, so it should not be LH. If we +2, it should be K. It's not become K. So definitely it is not the right answer. HL or LH is not the right answer. So we need to think of it differently. How? J +1 K. Now let's assume they increase it to +2. K +2 LM. So it is M. Then +3 NOP. Good. Then +4 QRST. On the other hand, again increase them, D +2 F +3 I +4 M +5, sorry, yes, +5 R. So it will be MI. So you answered wrong. Bulk. In bulk you said HL or LH, whatever. Think about it a little carefully, okay? Because sometimes what happens, even though you know how to solve these questions, rushing these questions can lead to the negative markings over there. And you will confidently tell that it is HL. But it's not. Okay. Let's move to another problem. BD single one, GKP. This one, BDGKP, and then what will be the next? Very similar to the previous one, and it's easier. Anyone with any thought? If there is a single line, there is only one way, addition or deletion. As we see D after B, so definitely there are additions. So addition means what we are talking about here. We have different results. We have U, P, V, I don't know what is, so let me do that. B we have plus 2, so we get B. Then after G, so D, E, F, G, so plus 3. After G, H, I, J, K, so plus 4. So easily we can see it's going like plus 6 of P. What will be plus 6 of P? V, right? So the answer will be V. Great. Another one. Another one. MR, PO, SL, VI, then what? Then tell me this one. Again combinations. So break down the combinations. M and P. So obviously there will be addition here because as we are going in the upward directions. M plus MNOP plus 3, right? QRS plus 3 again. TUV plus 3 again. So plus 3 after V. Y. Good. Shweta said YF. Let's see in the next round. That is ROL. So what we are going downgrade. So obviously something negative is going on. So deduction or deletion will be going on. So R to O, OPQR, so minus 3. LMNO, so minus 3 again. JKL, minus 3 again. And 3 minus from I. GHI. Or F. So the value will be YF. That's correct. Okay. Now with some numbers. Let's say 5, 7, 11, 20, 35, 67. Now find out the odd one. Odd one out. This is the odd one out series. What is your thought on this? Why? Why 20? The pattern we are seeing here. All are odd, 20 is even. Why 35? It's a very easy one. Don't complicate it much. Yes, it's 20. Clearly 20. Okay, and that's very basic. If you think of all odd and even, this is 1. 20 should not be there. Now if I ask you what should be there, then what will be your answer? What should be present there instead of 20? Exactly, Vandana, it's 19. Very good. Should be 19 instead of 20. That's good. Now the next one, 4, 11, 21, 34, 49, 69, 91. Same type, odd one out series. Rushita said 11. How? Okay. Anyone else? Question is again, you need to find the odd one out. 49, Vandana said why 49, and if 11, then why 11? There will be the explanation for it. What is the explanation? Look at it, the numbers are increasing, so obviously it's the addition, just like the alphabets. If you add 7 with 4, you get 11. Then if you add 10 with 11, you get 21. Then you add 13, you get 34. Then the difference again is 16. No, not 16. It should be 16. So it should be 50, because after that, because if it's increasing plus 3, every case it will add plus 3. So now it will be 19 plus 50 will be 69, and 22. What happens in plant cells, the nearby cells start to shift their cytosolic contents from one cell to the next cell, and they are transferred via small chambers and chambers that are created between two adjacent cells. So that is known as the cytosolic streaming, the plasmodesmata, which could be either in case of plasmodesmata or in case of the movement of a cell. Let's say a cell is moving towards let's say a food source or chemical source for a signaling type. In that case also cytoplasmic streaming is very important for the cell to keep moving in a specific direction. Okay, now in this case, the cell division also, it is another thing, but in the less aspect for the microfilaments. Okay, the actin filaments plays less important role there, but most of them plays an important role, like I told you other things. Now microtubules are made up with tubulin molecule. The protein unit that produces microtubules are tubulin. There are 3 different types: alpha tubulin, beta tubulin, and gamma tubulin. The major structural constituents of these tubulins are alpha and beta tubulin. The gamma tubulin has different functionality of arranging them or not. The functionality of microtubules, the major function is the cell motility and the organelle movement, and obviously the chromosome movement during the cell division. These are the major 3 roles of microtubules, as you know. Now the structure of microtubule, if you look, it is far more complex compared to the actin or microfilaments. It is in this case the tubulin dimers. Now alpha and beta 2 tubulin will be attached together to form a tubulin dimer. Such tubulin dimers will be added one after another, and what they will do actually, they will form a plate, a sheet like structure. They will form a sheet like structure. Now once they form the sheet, then this sheet will be folded to form a hollow tube. Hollow tube means there will be a gap, the tunnel like thing in between. So they'll form this hollow tube like that. Okay, by wrapping them together. This is the idea. The column is produced by microtubules, and the depth of this, the diameter is very higher, like 25 nanometer, compared with the actin, which is 7 nanometer only. It's much bigger. It's much okay. the diameter is much higher. Now the third kind is the intermediate filaments, and the name suggests it all. Intermediate, why intermediate? Because the diameter of this filament lies between the diameter of actin filament and the microtubules. Okay, so in between something, like in this case, as you see, it's not I don't think 8 point something nanometer. Okay, so in this case, this is completely different. Just like, the example is keratin fiber. You know what is keratin, right? You all know, I guess. Keratin is a protein, fibrous protein. We'll talk about keratin when we talk about the module 1 part a little bit, because in module 1, keratin and collagen, these are the two proteins that you need to know in details, along with hemoglobin in the globular proteins. Now here, they produces this thin polymer. This multiple polymers will be twisted together, just like a rope. It's nothing but a rope like structure. Okay, it gives much structural integrity. Okay, that is the idea for the intermediate filaments. So three types: microtubules, the density, the diameter is the highest, then the microfilaments lowest, and intermediate filaments ranging in between, and the rest of the things provided. Okay, now let's talk about the structure in a little bit more details. Let's talk about them. We have separate slides, for each of these cases. For each of them, I have a separate slide which explains each of the structures in details. So what I am going to do, I'm not going to explain here because I have animation to show you, but this is for your understanding, because just take a printout of the first page. If you take a printout of the first page, it will be sufficient, because these are the same things explained again and again. Okay, so let's talk about that. Okay.
You carry out
Before that, let's talk about actin filament first. I hope everyone can see the screen. Just let me know once so that I can start. Okay. In actin filament, just try to see the actin filaments. They are known as the G-actin. In this case, because in this case, the addition of actin proteins, there is some idea. It is known as called nucleation. Okay, nucleation means once multiple molecules, once the actin fiber is a polymer made with actin protein. Okay, so there will be multiple actin proteins joined together to make the polymer. So before the start of this polymerization, what happens actually, small actin molecules start bombarding with each other, and what they do actually, meanwhile, once they are constructing from one end and destructing from another end, during that time they need at least 4 or 5 actin molecules, actin proteins, to come close to each other and attach to each other. Once that thing happens, it is known as the formation of the actin nucleus. Once the actin nucleus is formed, then only the new actin monomers will be attached and they will start. So you see here, this is known as the ATP actin, because ATP when attached with actin when attached with ATP in a specific location, it is in the active format of the addition, and ATP when we hydrolyze, it is known as the ADP actin. So there are two forms of actin. ATP actins are required for joining with each other, and that is known as the spontaneous nucleation. As I told you, they need at least 3 or 4. Continuous bombardment is going on, and at that time at least 3 or 4 actins, when they are added with each other, they will form an actin nucleus. Once the actin nucleus is formed, then so many actin monomers will be joined together to form what is known as the actin polymerization. Now remember here, two things you can see. As you see at the beginning, all of the actins are yellow. Yellow means the ATP bound actin, that is the active part of the actin. Once ATP bound actins are there, they will interact with themselves, they will fuse with themselves, and then start to form the polymer. Now as they are forming the polymer, as the chain is growing, there are two different ends that are formed. One is known as the minus end, other one is known as the plus end. There is nothing to deal with the charge here. Plus end means where the actins start to assemble, while minus end means the disassembly end. This is the end where, after arranging some 10 or 12 monomers of actin to make a polymer like structure, the earlier actins, they lose their ATP. They hydrolyze their ATP, they form ADP actin, and ADP actin will no longer be stable, so they can be separated, they can be released from as a destruction unit. Okay, so this is the idea. This is how the actin, as you see, the dissociation takes place from the minus end, the association is going on at the plus end. So this is the idea. So if you look at this specific way of arrangement and depolymerization, what idea you can get? The idea is this actin fiber is dynamic in nature. That means it is forming from one end and it is destroyed from another end. And that is the idea, that is the thing you required. Every time inside the cell, actins are arranging and again disassembly. These are the things that are going on every time. So this is the basic idea of actin polymerization. Now along with actin polymerization, we also talk about two drugs and their effect in the actin polymerization. One is the profilin, proteins actually. Profilin is a protein, and cofilin. These are the two types of proteins here. Profilin and cofilin, they have a specific functionality in the arrangement of this polymerization. Now profilin inhibits the nucleation. As you see, the specific ends which are required for the nucleation is blocked by the profilin. Once profilin is attached to the actin monomer, it will not allow the actin monomers to form the nucleus. And profilin always binds to the ATP attached actin, so obviously it will block the process of polymerization and start point of the polymerization even. Now the second part is the cofilin. Interact with themselves. Cadherins will hold their structure very closely together in a specific area of the cell membrane. And not only these cadherins are present outside the cell in the intercellular matrix, while the cadherins are attached to a specific point of the cell membrane known as the plaque, and the plaque contains other molecules like intermediate filaments forming in, like keratin. Now intermediate filaments like keratin will be also formed from the plaque, which are present in the intercellular, inside the cell, and cadherins are present outside the cell. So in this plaque is the area where something coming out inside the cell, something's coming out outside the cell. The outside elements are known as cadherins, inside elements intermediate filaments, example keratin. Now by this fashion, what it can do actually, it can interact with the nearby cells completely with a very tight interaction. Okay. Now this is the third example, again from a different view. You see the tight junction means these are the areas, how tight junction looks like. This is if there is no tight junction, how it looks like there. Okay, the molecules can easily pass through between the adjacent cells, and there won't be integrity between the cells. If you drag the cells in this direction, it will be spread across. Okay. So there will be no control in the absorption of food, because if you consider the same thing, the same tissue lining in the intestinal wall, and if there is no tight junction, there will be no control of the movement of the food. Won't be any control, because the food is absorbed through the cells. So if there is gap between the cells, there is no way they can move. Okay, the movement will be destabilized. What is keratin? I told you keratin is a protein that assemble themselves like an intermediate filament fashion. It's a fibrous protein which is present in different areas of our body. Can you name one? Can anyone name one example where the keratin is present in human body? Hair, yeah, it's very common, because now it's in every shampoo, I guess. In hair as well as in nail, both the area are, yes, the construction of the keratin. It provides very
Desmosomes are intercellular junctions which anchor neighboring cells together. Each cell produces proteins known as cadherins that extend from the cell and anchor to the cadherins from the neighboring cell. There are different types of cadherins expressed in different tissues. The cadherins stem from plaques on the cytoplasmic side of the cell, where they bind proteins known as catenins. There are a number of anchoring proteins which can exist in the plaque. So, for example, beta-catenin can connect the cadherin to alpha-catenin, which then attaches to proteins of the cytoskeleton, such as intermediate filaments like keratin or perhaps actin. And thus the cytoskeletons of neighboring cells are joined. And if stress is placed on one of the cells, such as in the skin, or as cardiac muscle cells contract, this stress is not only passed on to neighboring cells to anchor them together, but also distributed evenly throughout the cytoskeleton of each cell.
So you see why it is important to form a structure like desmosome,
because it helps the cells to maintain the integrity as a whole, not as a single cell, but as a unit of one cell that is distributed across the other nearby cells, which will be much beneficial to fight the stress. Is much easy to be distributed properly. Another thing we must talk about is plasmodesmata. I talked to you. To maintain the balance, the idea is the same in diffusion and osmosis. In both the cases, the equilibrium should be reached, and the equilibrium can be obtained if we have a same concentration in both the sides. But in case of osmosis, one molecule is prevented to move, so the other one moves. So osmosis can be termed as diffusion of water. That's it. So water moves from high water content to the low water content. But in that case, is there any role of hydraulic pressure in the osmosis? Not a role in the pressure. The pressure, the movement of water depends on to fill this idea of same concentration. To make the same concentration, water flows, and when the water flows, it creates some pressure, some hydraulic pressure, and that pressure could be of different type. If you look at in case of a cell biology, that pressure could be a target pressure. In case of plant cells, it can act as a target pressure, and that pressure is entirely built by the water. That is why you see, if you take some green plants like that, they will see the very rigid structure of the plant. If you put them in little bit of heat, water will evaporate, and you will see there will be a flaccidness in the structure of the plant. And again, if you dip them in water, you'll see the plant will gain some water, and it will get that. I can't do anything because I am talking about the stuff. If you can't hear, just rejoin in these cases, or you can hear them in recorded class. Now another thing I want to talk about is the water balance in the cells. Okay, cells without the walls and with the walls, both the things we'll talk about. The water balance is very important. It is known as osmoregularity. That is the water balance between the cells. Otherwise the cells can die even if there is a difference in the concentration gradient across the cell. So there are three things I will talk about. Tonicity. Tonicity is the ability of surrounding solution to cause a cell to gain or lose water. That is a term called tonicity. Now there are three different type of tonic solution that are possible. One is the isotonic solution, where the solute concentration is same. Let's say let's go to the example straight. There are three situations. One is the isotonic solution. Isotonic solution means the concentration of the solute that is present inside the cell and outside the cell are same. That is known as iso. Iso means similar. In isotonic condition, what happens? The concentration of solute same inside the cell, outside the cell. So what happens? Water molecules will pass. These are the water molecules shown there. Water molecules can come in and out, but ultimately it will not provide any net gain or lose of the water. As the cell is present in isotonic solution, where the concentration of the particle is the same in both in and out, they will not cause any net gain of water or net lose of water. So the cell remains as it is. It remains its structure integrity inside the cell, inside the solution. The second type are hypotonic solution. Hypotonic solution means the cell contains, let's say, a particle glucose or let's say sodium chloride. The concentration of sodium chloride inside the cell is higher compared to the outside the cell. Less amount of sodium chloride outside, more sodium chlorides inside. So what will happen here? Try to think of the idea of the osmosis. The cell membrane is acting as a semi-permeable membrane. It will not allow sodium chloride to pass. It will only allow water to pass. So as the content of sodium chloride is higher inside, the only way to make the concentration even, water should come inside the cell. So there is a net inflow of water inside the cell, and it causes the cell to swell up, and it may even cause the cell to burst. Okay. The third case is a hypertonic solution. Hyper means more, hypo means less. Hypertonic means the solution contains less amount of sodium chloride where the cell is in, and the cell contains high amount of, sorry, the hypertonic means the outside the solution contains more amount of sodium chloride and the cell contains less amount of sodium chloride inside. So what will happen? Sodium chloride concentration is higher outside the cell, lower inside the cell. And again it is separated by a semi-permeable membrane, which is cell membrane. So sodium chloride will not pass, it will not change, it will not go from outside to the inside. Only water can move. To make it in balanced condition, water tends to move from the cell outside the cell. There will be net outflow of the water. It will cause the cell to shrink. Okay. In this case, we see the example with the red blood cells, and if you do the same thing for the red blood cell, there is another example. If you put the red blood cell in isotonic solution, no net inflow, no net outflow, so the cell will behave normally. If you put them in hypotonic solution, water will flow in, ultimately it will break down the cell, burst open the cell, cellular contents will be released. And if you put them in hypertonic solution, cell will release water, it will be shriveled, it will be dehydrated. The same thing if it occurs for a plant cell, what will happen? Because no matter what condition it is, plant cell membrane is again further surrounded by cell wall, which is very, very rigid. So the scenario is little bit different. Even if the water is losing from the cell will not be completely shriveled like that. It will maintain their structure, but the structure will not it won't be that strong, but still it will try to make its structure a little, it will be less rigid, but it won't be that bad. Okay, so what happens here in plant cells? There is a scenario like when if you put them in an isotonic solution, the structure will be flaccid. Flaccid means normal. The structure, it won't be very much strong enough, very less enough, because there will be no net inflow, net outflow. Now if you put them in hypotonic solution, the same thing, water start trying to rush in. Water try to rush inside the cell. If water rushing inside the cell, water will create immense pressure on cell membrane, because cell membrane and then cell wall. So water is creating immense pressure in the cell membrane towards the cell wall. This pressure is known as target pressure. Ligands or frog or chicken, and sometimes human, but very less. Okay, sea urchin is in fertilization. This is from sea urchin. This is the part they ask from sea urchin, the fertilization part. Gastrulation mostly from frog, because we studied the gastrulation in frog in much more details. Drosophila for the morphogenesis and body segmentation pattern, and it can ask from some part of the C. elegans life cycle and C. elegans structure related thing, frog, chicken or human. Even if they ask from human, it's very less, but mostly they'll try to ask from different regions. Okay, that is the idea. So let's start with this part, and if you study this fast, video that we're going to study, this is going to clear your concept about the fertilization as well as gastrulation. These two things will be very clear. So I will tell all of you to follow this part, because if you answer 2 questions from developmental biology, that will be an added advantage to all of you. Okay, so let's go. And actually, frankly speaking, from the questions of this developmental biology, they are not very flat all the time. I mean, even if you read some part of it, it's not even enough to answer all the questions. Okay, so what you can do is that you can expect to answer 50% or 60% of the questions that you get from the development biology, not most of that, because we can't study in very detail about the developmental biology right now, because the idea is, if you want to study developmental biology, is a whole new world for all of us, and this world, if you want to study for each and every individual of organism for their whole details, like from the fertilization, gastrulation till the end, it will be very hard for you to remember all the stuff. And even if you do that, again, the issue is about the economy. That is, once you study, it will take almost 3 months, for example, for your hard work, it will take 2 to 3 months of your practice, and you can maximum get 4 to 5 questions to answer. Okay, so this is the thing. So for that reason, we'll study the basic part very well, so that applying some knowledge in some experiment design type of question we can answer, and we'll go for gastrulation and fertilization in much more details. Okay, now today's class will talk about the fertilization and gastrulation in details. In the later classes, we'll talk about the modularity and a little bit about the C. elegans. Okay, so let's talk about, so what is development? Development is a series of progressive changes that generates the structure of an organism throughout its life cycle. Okay, as I told you, it's an accumulation of small changes over the time period. Okay, so for example, see, it includes the growth. Let me erase all the stuff. Erase all the stuff. I think this is eraser. Yes. Okay. I think you can see that. Okay, it's erased all the things. Sorry. Anyways, I'm explaining that in this format. Okay, the development includes 3 things. It includes the growth, that is the change in size and increase in the cell number. So growth in the 2 form, the cell size is increased as well as the number is increased. Okay, that is called the growth. It involves in the cell division. And okay, second thing is the differentiation, which is the cellular specification. Okay, so specific cells will be allotted to do a specific work only. Okay, this is the differentiation in their work. And the third thing is the morphogenesis, which establishment of the body pattern. That means how the body will look like, how the shape of the body will look like, and all those things. These are the 3 changes that we see in a development: the growth, the differentiation, and morphogenesis. Now if you see, a simple example is provided here about the starfish. All know about starfish. It's fascinating in looks. If it starts with a zygote with 2n number of chromosomes, and then start dividing, produce 8 cells, and then it produces different forms. I will talk about each of the forms in much more details later. Forms, first it's the 8 cell stage is called morula, then it's called the blastula, which forms a hollow shaped ball. Then it forms gastrula. Gastrula is a phase where the differentiation taking its toll, and it's making the cells at a specific type of cell going to a specific directionality of their function. Then it goes to a specific larval form, then it comes to the adulthood. So you see, from a single cell it produces an organism with different sequential events. Okay, now as you all know that as it's a single cell originating a whole organism, definitely there is something to deal with the cell cycle. Obviously cell is dividing, it's required, and not only cell division but also different types of changes. It's a game of changes, cross play between the genes and the gene products. Okay, now one thing I should say about the differentiation, is that determination precedes differentiation. So what these two terms means, determination and differentiation. Determination means commitment of a cell to a particular fate. Okay, for example, a cell is specifically fixed to produce neuron. Okay, a cell is specifically designed to produce digestive system. These are known as the determination for the cells. Okay, once there is only one cell, there is no such determination. Every task, everything, every gene and component is present inside that cell. But when they start dividing, some of the cells are allotted to produce digestive system, some of the cells are allotted to produce respiratory system. So different cells are allotted to do different tasks. Okay. Now this determination is affected by two things mainly. One is cytoplasmic segregation, induction. These are the two things. First is the cytoplasmic segregation and second is the induction. What does these two things mean? Cytoplasmic segregation is the cell division simply. Cell division determines the type of determination a cell will proceed. And second is the induction. Induction, what is induction? So let's talk about induction in a little bit more details. Induction is where one type of cell influence another type of cell to produce a different functionality. For example, this is a process in which substance or tissue influence the fate of group of adjacent cells. For example, just look at this example very clearly. Now this is the development of a frog brain and lens. Okay, so say this is the frog head and this is the developing forebrain of the frog. Okay, these are the tissue lining that we see out there, specifically determined tissue to produce the brain cells. And there is a layer of external layer that is present outside. It's the external layer there. Okay. Now these cells, if you zoom into this portion, you see that this head or the brain cells that are present here, they start to align in a specific structure. Okay, this alignment will ultimately force the surface tissue to get a specific structure. What is the structure? It is the formation of optic cup. Okay, as you see, it looks like a cup. The formation looks like a World Cup, thing like that. Okay, it's forming like a cup, and once forming like a cup, it influences this lens placoid tissue to involve in the same type of alignment. Okay, it's influencing this. Once it is providing the structure in this opposite end like that, it is forcing it. But now as it is forming this cup like shape, it is allowing this lens placoid to form engulfment like that. And slowly these two regions of the tissue will come in close and they will be fused. Once they are fused, they will form a lens. That is how a lens is formed. And you know, this formation of lens is influenced by the presence of this developing forebrain tissue that is present there. Okay, and after the formation of the lens, this lens will finally implement or add other layer of tissue, cell layer that will form the cornea. Okay, so this is how the induction works. Okay, now this is the simple idea that we talked about, the different terminologies that we talked about, that what is the determination and what are the different stages of the development that should be done. Now we talk about from the beginning, that is from fertilization. We'll see how a cell slowly modifies itself ultimately to produce an adult organism. Okay, so this is the fertilization step, and the fertilization step, the first step, this is the first step towards the development, and this is the contact between the sperm and egg shell. Okay, this is the first thing. And second stuff of the fertilization is the contact between the sperm and egg's plasma membrane. Whenever they will be fused, the nucleus will also be fused and close together. So let's talk about the fertilization. And you know, in developmental biology, the study is not limited for one or two organisms. Okay, we need the model organism for the study. And what we know is that in developmental biology, we study different situations or different events in different organisms. Okay, for example, we study the fertilization event in sea urchin. because it is easy to study in the sea urchin. We study the gastrulation process more in frog and chicken, but it's more in frog, okay, to understand that event as a unique feature. And we study front of the process of known as acrosome, okay. Acrosome means this is a specific section of the sperm head where they have a lot of Golgi bodies. The structure of Golgi bodies are filled with vesicles. These vesicles are filled with different types of hydrolyzing enzymes, okay. Different hydrolyzing enzymes they are present there in the acrosome. So what happens is this acrosome contents are released. These contents are released in the zona pellucida, okay, and they'll start to destroy the zona pellucida there, creating the room. Once the zona pellucida layer is destroyed, then this sperm can come close to the egg membrane, okay, and then it can fuse. This is the idea. Then it can be fused and it can deliver its content inside. So let's talk about each of the stages in details. For example, the first stage of interaction between the sperm and the egg outermost side, and how this thing is done. So this is the egg and the sperm, okay. The sperm's acrosome, and in front of the acrosome there are specific regions, you know. This is known as a binding molecule. It's known as a binding. Just give me a moment to take the call. Hello. Try to restart, Ramesh. There's some problem because other people joined. Problem. There's no problem with it. I think there is a problem. Okay, so there might be a problem related to you just turn on and off your internet connection and restart your system and try again, okay. Okay, so what we see here is the interaction between the egg and sperm head is between a specific molecule known as binding. And if you remember, in December they asked a question from this area about the binding molecule. Binding molecule is present in the surface of the sperm head there, okay, and the binding receptor is present on the surface of the egg, okay. So this sperm can interact with the egg with the help of this binding and binding receptor interactions, okay. So binding molecules will interact with the binding receptor and they'll be attached with each other. This is how they interact with it themselves, okay. So see here, the step-by-step process is provided. So acrosome. So normally you see the structure of the sperm head. It's not that much thin at the front, okay, or it's not been pushed outside. But what's happening here is that they have actin molecules, the presence of actin molecules out there, monomers of actin. Now what happens as they come very close to the vitelline envelope, this actin start to rearrange themselves, okay, and then form the actin polymer, and this actin polymer will push this acrosome contents. They will push all these vesicles and contents to the forward direction. So as they are pushing it to the forward direction, then acrosome contents are released outside and they will degrade some part of this jelly coat, and then finally this sperm will ultimately interact with the membrane, and once they interact with the membrane fusion will take place, and then the nucleus will be taken inside, okay. Okay, now if you look at the example number 2, that is in mammals, what happens in mammals is a little bit different. That you see here, the Z, there are different types of, you know, I talked to you about the zona pellucida, is the structure of vitelline membrane, the zona pellucida. The zona pellucida contains different types of carbohydrate molecules, ZP3, ZP2, ZP1, they are the different types. So what happens once the acrosomic reaction takes place, Once the acrosome reaction take place, that is the degradation of all these components. So you see that ZP1 and ZP2 are cleaved. Only ZP3 is present. 2 and 1 are cleaved. So after this cleaving of the ZP1 and 2, this membrane integrity is destabilized. The integrity of the membrane is destabilized, and as a result of that, what happens is that water start to flow. In case of sea urchin, if we talk about, or water start to flow inside. Okay, so as water start to flow inside, it swells up the egg structure. It swells up the structure, and this can prevent the further fusion of sperms. This is a very secondary approach for blocking the polyspermy. There are some major approaches. We'll talk about that later. Okay, so this is the idea. They will break down the extracellular matrix component, fusion is established, and the nucleus is delivered. Okay, we talked about it. Okay, now if we talk about the polyspermy prevention, there are two types of polyspermy prevention. One is known as the fast block, which is also known as the transient block. That in this case they prevent the sperm fusion only. And second is known as a slow block, and it is a permanent type of block where they remove the bound sperm. So what happens actually, if the egg is there, some sperms can be bound and some are not bound. So in fast block they prevent the unbound sperm to be attached, and the second slow block they will remove whatever sperms are attached. Okay, these are the polyspermy prevention. So this is a chemical signal. Remember, cells when you start to migrate in the surrounding regions. Okay, cells slowly start to migrate there, and they form a sphere. That means they form a hollow sphere. The thing that is present in the middle here, this is known as the blastocele. Okay, blastocele, and the cells of the blastula phase are known as blastomeres. Blastomere means each cell. One of the single cell is known as blastomere, and blastocele is the hollow chamber that is created. Okay. You see the example of discoidal cleavage in fish and birds. You see, this is the egg on the top. The division begins, and divide, and they'll form a bunch of cells on the top like that. Okay. What counts the pattern? What controls the pattern? You know, the second thing we talked about the factors, that is the angle and timing of the cell division and spindle formation. Now, in this case, you will see the idea of how they will arrange all the stuff. For example, there are two types of organisms: protostomes and deuterostomes. Example of protostomes are mollusks, annelids, arthropods. Okay, and deuterostome examples are echinodermata and chordate. Okay, they belong to this. So the idea is, in a protostomes, what happens? This is, let's say, this is the blastomere. This is the blastula phase. Blastomere. The blastomere means each single cell in blastula phase. If you take one of the blastomere out, it does not involve in any change. It involves in the change. Like if you remove one blastomere, the rest of the other blastomeres, those cells will not be able to grow and divide. So the development will be arrested at this point. Okay. But in case of deuterostomes, even if you remove one of the blastomere, rest of the cells can easily develop normally. And the single cell that we also have, they will also develop normally. Okay. So it is depending on specific type of cleavage pattern. For the protostome, they belonging to the cleavage called spiral cleavage. For deuterostome, they belonging to the cleavage called radial cleavage. This is one thing. The radial cleavage and the rotational cleavage, this is one type of thing. There's a radial means a straightforward cleavage. Spiral is the orientation. Spiral cleavage, you see, radial means if this is the first round of cell division, the second round of cell division, the cells will be originating in the same direction. Third round of cell division, let me take another color, red, same orientation. But in spiral division means this is one type, the second type will be different. As you see in this picture, it's much more clear. Okay, this is a spiral. In the third round they will again go like this, fourth round they'll again go like that. This is how they will construct the layer of cells one after another in spiral. When in radial they construct the layer like a stack in the same fashion. Okay, now in case of deuterostome, remember the idea is, if you look at the blastula structure, blastomeres surrounding blastocele. This is blastocele, this is our blastomere, and at the start point of the gastrulation there is a small pore created in this chain of cells, small pore. That pore is known as blastopore. Okay, and remember this blastopore that is created here, this forms into anus in case of deuterostome. But in case of protostome, this blastopore turned into mouth. These are small informations that might be important for a group B question. That's why I'm telling. Okay. And the question is why they belong to that such kind of difference. Like, you know, as I told you, like if we drag one of the cells away, in protostomes they will not divide, but in case of deuterostome they will produce a healthy organism. The reason behind that is the cytosolic components. As I told you, that there are specific protein components required for the cell to grow and divide and ultimately mature to an adult organism. And this maturation requires those specific proteins present in the cell. Okay, in the cytosol. And that thing varies. That thing varies from time to time. For example, in case of this protostomes, in case of protostomes, those cytosolic components that we are talking about, they are not present in this part of the cell, so they are not functioning properly, they're not producing any organism. But in deuterostome, at the very early point of development, all those blastomeres receive some part of those cytosolic components. As a result, even if you spread them and separate them, they are eligible to go and form adult organism. Okay. Now about the radial cleavage, you know, radial cleavage means, you know, simple slices from the top or horizontally, either meridional or equatorial. Okay, in either way. It's not required to tell, but yeah. And the rotational cleavage on the other hand, I told you the rotational cleavage means it will be completely, the cleavage pattern will not be considered the same fashion again. It will be rotated every single time of the division. So the first, one minute. So here you see the cleavage plane 1. So they'll divide from the cleavage plane 1 first, then they'll use the cleavage plane 2. This is the second. Third is again an equatorial. Fourth can be again from the top. Fifth can be again from equatorial. So they will do that. They will do that and cross change again and again and again, one after another. So this is for the echinoderms and amphibians. This is the radial cleavage pattern we are talking about. It can be one straight, then again straight, third one again straight. They can change the directionality, but they will become belonging to the same type, meridional. But in case of the rotational, one will be meridional, another one will be equatorial. So first vertical, then again horizontal, again vertical, again horizontal. So this is the idea of the rotational cleavage in mammals. Okay, so now we are at the end point of blastula. Okay, like kind of midpoint of blastula. So once the cell division begins rapidly, we know that from the end point of the fertilization, cell only have two processes, synthesis phase and mitotic phase, to divide. But now once the blastula is being processing for a while, the cell decides to again shift back to the normal type of cell division phases. Okay, so from the S and M phase only, they belongs and come back to the complete variance. So it's G1, S, G2 and M, this type of phase of cell division, and this time is known as the mid blastula transition, because this whole process takes place at the middle point of the blastula phase. Okay, so blastulation, at that time of blastula phase, at the midpoint of the blastula, they decide to shift back to the complete phases of cell cycle. Okay, so again in this case you see, so in this case embryo start transcribing its own RNA, okay, and then the RNA start making their own proteins, so they can produce a lot more proteins, because you know, if they shift to the G1S, G2NM phase, they will require different checkpoint proteins like cyclin E, A, B, D, all these things. Earlier there are only one proteins required, that is cyclin B, which is present in the egg cell itself, okay, but now they require multiple proteins, and they have multiple protein function, that they have their own RNA to produce the multiple proteins, okay. So what derives this what drives this mid-blastular transition? Okay, the clear example is a nucleus to cytosol ratio. This is the contender of driving this whole process to the mid-blastular transition.
I told you that I'll start 5 minutes later. Okay, so let's begin with it. Now everyone can hear me, right? You guys can hear me? Okay. Today we'll be covering Group A. One last Group A explanation from the last year question paper. Okay. Because you know, the earlier that means we talk about 2015 June NET question paper of Group A we already discussed, and today we'll discuss about the 2013 June NET question paper. June or December, I don't remember exactly. I think it's a June NET paper for 2013 Group A discussion, because it will give you an idea of what type of questions you may get from Group A. And actually I told you earlier also, with your common sense you can answer some 3 or 4 questions from Group A. So do not just leave Group A blank. With your common sense you can apply and answer some 3 or 4 questions. And if you prepare some of the questions are tricky and difficult, so leave them if you don't want to handle those questions. But on the other hand, if you want to solve, if you have some idea about some formulas regarding the different 3-dimensional structures, you know, like cone or cylinder and stuff, then you can obviously answer 1 or 2 questions from there. So let's begin with this first question. The first question is about this one. Oh, 1 minute. Okay, so the question is about a 16.2 meter long wooden log. 16.2 meter long wooden log has an uniform diameter of 2 meter. So the idea is about, this is the wooden log, and you know the log looks like something like this, is cylinder. So diameter is 2. This is the diameter 2 meter, right? And the length is 16.2 meter. Length is 16.2 meter. So to what length the log should be cut to obtain a piece of 22 millimeter cube volume? So the volume we want should be 22 meter cube. Okay, that is the volume we want, and the length we know, and also the diameter we know. So once you know the diameter, you can get the idea. So 2r equals to 2 meter. So what will be the r equals to 2 by 2 or 1 meter, right? Now it's telling us that the volume, they provide us the volume, but they didn't say about where exactly we will cut. Okay, now let's assume that we will cut the segment x meter. Okay, we'll cut x meter. Yeah, I'm back. I'm back. There was a problem in internet connection. Okay, now you can hear me, I guess. You can hear me, right? Devraj, give the answer C. Very quick in calculations. Very good. I don't know. Let me check this answer first. So what the scenario is, it's easy to solve this problem. Let's begin as a starting point. Let's say the start point is here, and as they're moving south, so we'll take a start point on the north. If they start going north, then we'll take it in the bottom. Okay, so it's moving south for 30 minutes, right? Say A, this is the B, moving 30 minutes. Here it stays for 5 minutes, 5 minutes delay, collecting honey, and then it moves 40 minutes due to waste. So again waste 40 minutes, say point C. Well, everyone answers this, so it's okay. But let me again tell, because for other people, so they moves to C for 40 minutes, and there they stay for there, right? And collects honey in another garden for 10 minutes. So again stop for halt for 10 minutes. And then it returns to taking the shortest route is this one. As you all know, this is the shortest route. So how long was the beehive? 30 plus 40. So what will be the idea? 30 plus 40. Normally the distance 30 plus 40 plus 10 plus 5. But remember, the bee definitely took some time to come from C to A, right? Because this time they didn't tell us. So indirectly they want us to find the distance of CA. So how to find the distance of CA? The formula. What is the formula? Root over of AB square plus AC square, isn't it? So what are the values? AB is 30, AC is BC, AB square plus BC square, sorry. What is the value for BC? 40. So let's put the values in here, and so it's 50, right? So along with the values that we got, we need to add this distance, this time also, because 50 minutes are required for the B to come from C to A at the end, right? So what will be the value? 135, right? That is option C. Okay, is it clear, everyone? No audio. Is my audio clear to everyone? Yeah, so the problem is for I think most of the cases is clear. So if you have any problem, you can just rejoin. Okay, just cancel and rejoin. Okay, let me take another question. Okay, try to find this one. So this question anyone can answer. See, this is the easy question. So you need to look for this type of questions and answer them. Okay, otherwise it will be a loss to completely leave group A. Just focus on them. A bird, I think it's whatever it says. A bird is present at the top of a 15-meter high tree, and actually this type of questions I saw twice or thrice in the exam. So be prepared for this type of exam. It's actually height and distance program, height and distance map, but be prepared for it. So bird is present at the top of 12-meter high tree, sees a centipede moving towards the base of the tree from a distance equal to twice the height of the tree. Okay, the bird flies along. What is it again? I don't know. Actually, I used OCR to format this, and I think there are some issues with the. Let me check it. The question once. along a straight line. Okay, this is going to be A, supposed to be. So the idea is a bird is sitting on the top of a tree. The tree is 12 meter long, 12 meter tall, and this is the base. And what it is finding, that a centipede is crawling. Centipede is now present at a point. Let's say the point is A, and the distance between the centipede and the base of the tree is twice as the height of the tree, that is 24 meter now. Okay, a centipede is moving in this direction. Okay, so they are present and both of them have a same, what is telling us, the straight line to catch the, okay, the bird flies along a straight line to catch the centipede. If both move at the same speed, at what distance from the base of the tree will the centipede be picked up by the bird? So the idea is centipede is keep on moving in this direction. So let's assume the point X where the centipede get caught by this bird. So the bird is eating centipede at this particular point. Okay, let's assume this is as a B, C, this point is X. So this is the idea, bird is sitting here, speed is the same. Okay, bird is moving in this angle, centipede is moving at this direction, bird catches the centipede at this point X. So what we need to calculate? We need to calculate the distance between it. We need to calculate this distance. Okay, so this distance C and X, that means at which distance they will catch, the bird will catch the centipede from the base. So we need to calculate Cx, this distance. So okay, so let's assume the idea of height and distance in this particular case scenario. What will you find? It's let's assume this this Bx, as they are moving in the same speed, as both of them are moving in the same speed. Remember, after a specific time the centipede moves from A to X while the bird is from B to X. So if they take the same time to come from B to X and A to X, so we can say the distance between Bx and Ax are the same. Okay, this distance and this distance are the same. Okay, because they have the same speed and they are catching it at the same time. So let's assume it's as a y that we need to calculate. If we can calculate this Ax, this y, this value for y, then we can easily subtract it from 24 to get the answer, that is Cx, right? So Cx equals to Ca minus Ax. The value for Ax is y, that we know, we need to calculate that value. Okay, and Ca value is given 24. So this is our formula. We need to find the value for y, and we know what is y. Ax and Bx both are y. Okay, so what we can say, if this is the idea of Bx, if this is the case, so we can say like this: BC square plus Cx square equals to Bx square. We can tell that, right? So if we can say that, we know the values. BC square, the value of BC is 12, so 12 square. Cx, we don't know. Cx value is, you know, this is the value, 24 minus y square. Bx square. Bx is also y, right? Isn't it? If you don't understand, just pause me at that point, okay? Because until and unless you pause me, I don't know whether you are getting it or not. Are you getting it? You guys understand what I am doing here? The formula is very easy, just thinking about the stuff, okay? It would have been better if I take this point as a E or F something else, okay? Because usually people take X as a variable, right? I didn't take that, so that kind of really confusing. But if you take, you can take any other point here, okay? So this is the formula. We need to calculate Y here, so it will be 144. We all know that. Now the value that we need to calculate here. How to calculate the value? a minus b whole square. So we can put the value of a square plus a square minus 2ab plus b square, right? So square of 24, what is the square of 24? Just check square of 24. 7, 6, yeah, 576. So the value we can put 576 minus 48y plus y square, right, equals to y square. So what is going to be our value? 144 plus 576 minus 48y equals to 0. And now 48y equals to this value. What is the value? 4, so 580, 620, 720. So the value for y will be 720 by 48. What is the value? Just calculate. 24, if you multiply with 24, 2, 30, 15. Yeah. So we get the value for y is 15, right? So once we get the value for y is 15, we know the total is 24. So what is the distance between them? That is the cx, right? So cx equals to 24 minus y. 24, y was 15. So the value will be 9, isn't it? Where is 9? Option B, 9 meter will be correct answer, isn't it? Is it clear? Okay. Okay, let's take another one. There's an image to get. No. Okay. There's some mistakes. Okay. Let's take this one. I'm going to explain the scenario. It says an ant goes from A to C. Actually, there is a drawing. I didn't draw that. Let me draw this first here. Like this and this.